Contents of the lab report are meant for educational purposes only and not to be misconstrued as medical, diagnosis, or treatment advice.
Today, on the lab report, we're going to do Multi-Omics Part 4.
You dusted off the wheel, huh?
I did.
What do we got there? CBS, cystathionine-beta-synthase.
And it is dusty in here, man. Get a Swiffer.
The world of medicine can be challenging. Clinicians and patients are always looking for more options, more effective treatments, and in the end, more answers. Functional and integrative medicine focuses on addressing root causes of disease. Here at Genova Diagnostics, we've watched this field evolve and grow for over 35 years. We've not only adapted, we've led. Join us as we talk about functional medicine, laboratory testing, and optimizing health. Welcome to the Lab Report.
There's something about your face that makes me not believe you. It's like that charming Midwestern
Look,
face that makes me go, oh, something's going on behind that little
It's
little
usually
smile.
nothing. There's nothing going on. Hello.
Hey, Michael Chapman.
Hi, Patty Devers.
Welcome to the Lab Report.
I don't think that's a fair assessment of my face.
No, I'm just saying you have this Midwestern charming little smile, and I'm like, it's pretty sneaky behind that.
It's just that.
Just the
Midwestern
nothing.
charm. Just corn-fed Indiana boy.
That does not conjure up the image I have of you. Like, I think corn-fed Indiana boy, I think big farm guy, and you're just svelte and cool, collected.
Scrawny.
Yeah, like, is that because all you eat is corn?
It's monocropped.
Oh.
Anyway, welcome to this podcast put on by Genova Diagnostics, where we talk about functional medicine, specialty lab testing, integrative therapeutics,
And corn.
and just other stuff.
Yeah. If you're new to this podcast and you also like corn, you should probably go to iTunes or Spotify and subscribe to this podcast. Download, rate, and review. Leave us some stars there.
And if you would like to provide feedback, I'm sure you would after what we just did, you can email that to podcast@gdx.net.
That's right.
Exclude our supervisors on that email, please, so we can filter it appropriately. And what are we talking about today, Patty?
Well, you dusted off the old wheel, the Multi-Omics wheel, and it landed on cystathionine-beta-synthase. So
That's right.
that means we're revisiting methylation a little bit here.
CBS.
Yeah.
This is an interesting one.
Not the network,
Correct.
right?
Yeah, this is an interesting one. There's a lot of talk out there about CBS,
There
and
is. A lot of buzz.
man, I've even seen some barbs being thrown
I
around
have too. Very
about
true.
CBS in the functional medicine community.
People feel
which,
strongly about their CBS.
it's an interesting thing to get so passionate about, the biochemistry, but I can understand it because I enjoy it too.
Yeah, it's a very interesting place. But let's talk and remind everyone what we mean when we talk about multi-omics, Michael.
Yeah, so we've got all these big words, right? Proteomics, transcriptomics, genomics, phenomics, and they are really just the study of those things, the study of genes, the study of proteins, study of transcription, RNA.
So is like the study of books like bibliomics?
Could be.
How about sociology?
Sociomics is a thing.
See, are these just all made up words?
No, they're things.
Oh.
I mean, I don't know about bookomics.
Oh. Well, as it relates to what we're talking about here on our tests, we're going to layer the phenotypic and the genotypic markers together.
That's right. And when we do that, that's what we're calling multi-omics, right?
Mm-hmm.
And so that is what we're doing is we're looking at one particular SNP, in this case CBS, and we're saying, okay, what do we know about that enzyme, how it's functioning from a genetic perspective, and how is that manifesting in a phenotypic perspective by looking at the biomarkers around that enzyme?
And just as a reminder, again, you could go back to our prior episode where we talked about SNPs, or single nucleotide polymorphisms. And what we're talking about is just a variant in the genetic blueprint of that enzyme, which may or may not manifest phenotypically because we know our genes are not our destiny. So it's important to look at the genetic information as well as the phenotypic biomarkers around that particular enzymatic SNP.
Yeah, that's a great point, because that's the whole reason why we would consider doing a multi-omics approach, is because your genes do not make your destiny. They don't necessarily manifest, and therefore you need to look at the biomarkers.
All right. Well, let's talk about CBS, cystathionine-beta-synthase.
Go for it. What is cystathionine-beta-synthase? What does it do? Why do I care?
CBS, or cystathionine-beta-synthase, is actually the enzyme which converts homocysteine to cystathionine.
Homocysteine. Okay.
Right.
I'm familiar with homocysteine.
I am too.
We run this on a lot of our serum labs to get an assessment of cardiovascular disease risk. Why does it turn into cystathionine, and why does that make any difference to me?
Well, remembering that homocysteine is part of the methylation cycle, right? Remember that episode. And we know methylation is important for a lot of things. It's that reaction that helps us to detoxify toxins and hormones. It makes and breaks down neurotransmitters, immune function, a whole lot of really important physiologic processes, and homocysteine is part of that.
Yeah. Yeah, it's super popular because we talk about approaching methylation with various B vitamins, supplementation, folate, trimethylglycine, all these things to try and improve methylation and the methylation status of individuals from a detoxification perspective. Okay, so homocysteine is part of that.
Right.
So what does CBS do with respect to methylation?
Well, homocysteine itself sits at a very important branch point between the reaction of methylation and something called transsulfuration.
Yeah, okay.
So transsulfuration is the reaction that's needed to make glutathione for detoxification and also to feed the Krebs cycle for energy. Okay.
So I think about like a sink drain.
Oh, here we go.
So
Another Michael Chapman
there's water
metaphor?
in the sink,
Okay.
right? And it's that just kind of hangs out, and that's an idea of the methylation cycle. These things are going around, and these metabolites are going around and around.
In the
around.
sink.
In the sink. Yeah. And there's a drain at the bottom of the sink.
Okay.
And CBS is that drain. It's the plug. So if it's open just a little bit, if CBS is working at a certain speed, it's periodically draining the sink.
Okay, well, that one actually wasn't too bad.
Thanks.
So the importance of that CBS enzyme can't be overstated because it really is an enzyme that pulls homocysteine down at transsulfuration irreversibly.
And there's lots of things that govern how open that drain is, right? So you can have it open full stop, you can have it open part of the way, and genetics are going to determine that, as well as other activators, inhibitors of the CBS enzyme.
Right. And there are specific cofactors to CBS, things like vitamin B6 and
Which is
iron.
Going to impact how effective the drain is, how effective the enzyme is. Okay, so let's talk a little bit about the genomic aspects, the SNP aspects to CBS.
Okay. Well, remembering back when we talked about SNPs, there are two alleles. One comes from your mom, one comes from your dad. And if you have a variant in one, they call that heterozygous. If you get two variants, one from your mom and one from your dad, it's homozygous. And we also remember that to have a variant can either upregulate an enzyme or speed it up, or downregulate an enzyme and slow it down.
Yeah.
But it can also...
Or do nothing.
Right.
And there's not just one SNP as it relates to an enzyme. This is another important point, right? So we're measuring on the methylation panel, we're measuring one CBS SNP, and it's one SNP out of hundreds.
Yeah, there's hundreds of SNPs. We chose one called C699T.
Yeah, and we chose that one because there's some clinical evidence on it. It's documented. It's pretty well represented in the literature, but not without some contention either. And this specific SNP is really interesting because it actually does not change the protein structure of the enzyme.
Wait, what?
Yeah.
I thought SNPs, there's an amino acid substitution. Like, that's what the C and the T stand for, or the A and the T, that there's an amino acid substitution.
Can be. A lot of SNPs, there's an amino acid substitution, like, so you're building the protein, you're going along, and essentially how this works is when you're building a protein, it's like, here's an alanine goes here, here's a tryptophan goes here a methionine goes here, and you're taking these amino acids and you're building them together like building blocks. And so with a SNP, you're like, here's a tryptophan goes here, here's a cysteine goes here, here's a methionine. Oh, no, not a methionine anymore. Now it's an alanine or something like that.
Right.
And so what we're saying is that is oftentimes what happens with SNPs is a amino acid is replaced with a different one, which changes the actual protein structure. Sometimes for the better, sometimes for the worse, sometimes
Not at all.
doesn't have any effect.
Right.
But in this case, the actual SNP changes the amino acid from a tyrosine to a tyrosine.
That makes no sense.
Yeah, it's called a silent mutation.
Shh.
What?
It's a silent mutation. Well, if it's silent, why do we care? It must be making some kind of noise.
Exactly, right?
Mm-hmm.
You would think it has no effect on the enzyme's function. But it turns out that silent mutations can affect the enzyme because it can alter what's called their tertiary structure. It alters the way that they are folded and bent and their overall kind of conformation. So there is evidence to suggest that it can still have an effect. And in this case, the evidence suggests that a SNP at this location increases the activity of the CBS enzyme.
So it can potentially upregulate the enzyme,
That's right.
which is important because of where it sits in this branch point. So it would be irreversibly pulling homocysteine down into transsulfuration at the expense of methylation.
Yeah, so if you think about this drain analogy, then it can effectively open the drain a little bit wider, theoretically, such that metabolites, homocysteine, is flowing down into transsulfuration at a little bit of a faster clip.
And you might think, okay, well, great, we're clearing homocysteine, but if you remember, we do need homocysteine to keep the methylation cycle moving as well.
And there's always a set point. There's always feedback loops in these systems, right? So we'll talk a little bit about that in a second. But I think it's important to note that there is some controversy around this. And there has been some speculation that in some populations, there's actually an downregulation with this individual SNP. And so, it goes back to maybe this SNP is actually tied to other SNPs. Maybe there's what we call a haplotype effect where this has been compensated for, maybe overcompensated for by other SNPs around that area. If you actually, it's interesting, if you go and do the nerdy digging in DB SNP, database SNP, which is by the NIH, and look at all the individual SNPs that are in this gene around that location, they're all over the place. Yeah. It's crazy. So there's a lot going on right at that location in the gene, and it's population specific. So there is.
And we've talked about that before on this podcast, that, depending on what population you're studying, SNPs can do different things. So that's an important point.
Yeah. And so it goes to show that, again, if you have a SNP, you can't always say, oh, you've got this CBS SNP, your drain's wide open and you're draining all the metabolites. You have to look at the metabolites to make that assessment, to complete the story. Okay, so since we're about to get into the metabolites around the CBS enzyme and the CBS SNP.
Okay.
So it turns homocysteine into cystathionine.
Right.
Let's talk about cystathionine.
Great idea. Let's do better know a biomarker.
Hold on, let me hit this button real fast here.
Oh no, what button?
You think you know? You think you know your biomarkers? You don't know. You don't know your biomarkers. This is Better Know Biomarker.
Wow.
Yeah?
I don't know what to say.
Say about what?
That's got to be the most ridiculous segment opener you have yet. I love it so much, I don't know what to say.
Yeah, I mean, there's a lot of amphitheaters that are just available now to record these sort of things. You can get a rock band for pretty cheap. So
Wow.
So let's talk about cystathionine.
Well, my problem is now I'm afraid it's not going to live up to the hype.
Oh, it will. Cystathionine is a big deal.
Well, cystathionine, as you recall, is the end product from when homocysteine gets acted on by CBS, turns into cystathionine.
Okay.
So in this episode, we think, all right, well, let's talk about cystathionine. But if you remember, cystathionine then becomes cysteine and then goes through transsulfuration. And so cystathionine is really just an intermediate step in transsulfuration.
Okay.
So the problem is there's really no source or known physiologic function to even have cystathionine other than to be an intermediate in this transsulfuration pathway.
So it's relatively inert, is what you're saying.
Yeah, you would think, and you're like, well then, why are we paying so much attention to it? And I think importantly, number one, we look at it to kind of look and see what CBS is doing because it is that next byproduct. But additionally, there is some literature to suggest that cystathionine might actually protect against endoplasmic reticulum stress-induced tissue damage.
Really?
And cell death. But
Well, oh, okay.
But-
and that.
But the studies are sparse. There's not a lot of literature because really no one really cares about cystathionine apparently. But maybe they should start paying better attention to it.
Yeah, and we're going to start talking a lot more about this concept of ER stress.
Mm-hmm.
It's really interesting. However, in the meantime, yeah, you're right. So cystathionine is related to this entire process because it's the byproduct, the end product of CBS and conversion of homocysteine. So if you're looking at it from a multi-omic approach, cystathionine is going to give you information about if you have a CBS SNP, how fast CBS is working, especially how that concentration of cystathionine relates to like your levels of SAM and SAW and how that's balanced from a methylation cycle perspective.
I do want to point out, though, that not all elevated cystathionines mean CBS SNP.
Yeah.
Because downstream from cystathionine, there are many steps through transsulfuration that require other vitamin and mineral cofactors, things like B6 and zinc and other things like glycine and cysteine and magnesium. So if there are lack of cofactors downstream from cystathionine, it could potentially elevate cystathionine for that reason as well.
Yeah, and especially it's a very B6 dependent pathway.
Correct.
So a lot of times when cystathionine is elevated, we think about that. The other thing to think of too is what are the activators, inhibitors of the CBS enzyme? What are some things that are going to drive CBS to create more or less cystathionine, right? And so if cystathionine, we just talked about cystathionine potentially being high when the pathway is all backed up due to lack of cofactors. But what else could cause cystathionine to be high from a CBS activation perspective? Maybe a CBS SNP, like the one we're testing.
Right.
But what else?
Well, we also know that SAM is also an inducer of the CBS enzyme. So high levels of SAM in the methylation cycle kind of signal to say, we're good up here. It's okay to open that drain a little bit and go down into transsulfuration to make glutathione.
And the other thing I think about is oxidative stress. Free radical exposure is also going to induce the CBS enzyme to produce more cystathionine from homocysteine because it's reacting to free radicals and is saying, look, we need more glutathione on board. We need to get this transsulfuration pathway moving, so get CBS moving. And that's kind of how another reason why you might see an elevated cystathionine.
Right. So not all cystathionine elevations are CBS SNP. There are all these factors to consider.
Yeah. Okay. So that's when CBS is, or when cystathionine is on the higher side. What about low cystathionine?
Well, if you think about it, if high levels of SAM induce CBS, low levels of SAM or a poor methylation status might want to keep things up into the methylation cycle and not be pulled down irreversibly to transsulfuration. So poor methylation status or low SAM would eventually result in a low cystathionine. But then also, if you think about what are the cofactors for the CBS enzyme, right?
B6 and iron.
That's right. So if you have insufficient B6 or iron, it's possible you have a lower cystathionine because that conversion isn't taking place very well.
Yeah, that makes sense. And so if we go back to our drain analogy,
Okay.
I'll try this one more time.
Go ahead.
So the drain is open at a set point, and that's the natural steady state of CBS. And depending on the availability of cofactors, how much oxidative stress there is, how much SAM there is, all these things are going to slightly open or close the drain accordingly. And so that's going to just gently affect how much of the homocysteine is flowing down into transsulfuration.
Well, you know what time it is, Michael.
Oh, what time is it?
It's time for question of the day. And I'll tell you that you were so successful with your introduction of the prior segment that I'm hoping you've fixed our jingle for question of the day.
Fixed it?
Oh, no.
Oh, have I fixed it? If you recall, the last time that we talked about this, you challenged me
I did.
to come up with a new jingle.
Yeah, they were getting stale and old and boring.
So let me just say, I've fixed it.
Here we go.
It's the question of the day. It's the question of the day.
Wow. It certainly is fancy.
Thanks.
I don't love it,
Okay.
but I also don't hate it.
All right.
It is fancy. I think that's the best word I can come up with here.
Okay, I'll take that as a win.
All right. Well, here is the actual question of the day. And thank you to whoever emailed this to us, podcast@gdx.net.
Yeah, thanks.
Well, some people say that a CBS SNP causes elevated ammonia levels or hyperammonemia.
I've heard that.
Yeah.
I've heard
Is that
that.
true?
I've never seen that in the literature.
Okay.
I don't know that that's something that's been tested clinically. So if it is something that somebody's actually run an ammonia level on people who have SNPs versus doesn't have SNPs, that would be really interesting information to have. So like email that to podcast@gdx.net. I am not of the belief that a CBS SNP is going to create such a dramatic alteration, such as to cause hyperammonemia. I think the urea cycle is going to be able to compensate for these subtle changes that would occur with the CBS SNP. So the system is used to handling a lot of upregulation in the transsulfuration cycle, which in and of itself creates more ammonia, and that's handled. And so I don't think that this genetic SNP is going to cause something that is so far outside the realm of normal physiology that it would lead to high ammonia levels. That's my personal belief in looking at the biochemistry and looking at the literature, or the lack thereof, I guess I should say.
Great.
Okay, so we've talked a lot about CBS, what it does, and kind of the ins and outs. But how do you, if you're wanting to evaluate CBS activity or CBS function in your patient, how do you do that?
Well, here at Genova, we have this amazing profile called the methylation panel.
It is amazing, isn't it?
It is, right? And, it's a blood draw and also a buccal swab to look for the genomic material. And we layer the genotypic information right on top of the phenotypic biomarker measurements. And within the midst of the methylation panel, we do measure the CBS SNP.
Yeah.
We also measure all the biomarkers around this SNP, like cystathionine, homocysteine, et cetera.
Yeah, so it's a multi-omics approach to CBS. So if you have a SNP, then you can look at homocysteine, you can look at cystathionine, and you can even zoom out a little bit and look at the overall balance between the methylation cycle and the transsulfuration cycle. What if you do find imbalances? What are some of the things that you might think about from a treatment standpoint to help address CBS dysfunction?
Great question. And I think, okay, we know that a SNP upregulates. And if you look at some of those phenotypic markers and you see evidence that, in fact, it is manifesting, one thing that I always start with is I want to look at the methylation cycle. I want to make sure to support methylation because it's so important. So I'm looking at, is there poor methylation status? Is there something I need to support there first?
Perfect. And if so, find the root cause. Is it a need for folate? Is it a need for B12? Maybe a little bit of choline, betaine, things like that to help assist the backup pathways for methylation. That's great. Okay.
The other thing I think about is we know that cystathionine, because we got to better know that biomarker, we know that it's just an intermediate dipeptide, and we know that downstream of that, it's used to then make glutathione or to feed the Krebs cycle to make energy. So if you have a CBS SNP and cystathionine is high, it might be helpful to kind of support those downstream pathways with cofactors to help clear it to make glutathione or energy. And that's things like vitamin B6, magnesium, zinc.
Yeah. And then the last thing you think about is the CBS enzyme is regulated by free radicals and antioxidants. So you want to look at how much oxidative stress there is on board and whether the CBS enzyme is compensating for exogenous free radical oxidative stress. So that's the last thing I'd be thinking about, and look at glutathione, maybe look at some of the other elements. You could think about some of the markers on a Nutrival like lipid peroxides or 8-OHDG, which are going to tell you about oxidative stress damage, and that might be something to think about. Right, so what we're getting to is you don't just treat the enzyme, and you may not even just treat the homocysteine or the cystathionine, the biomarkers around the enzyme. You got to zoom out. You got to look at all these factors that regulate the CBS enzyme. So it becomes more of a complex assessment, but at the end of the day, you're getting to a biochemical root cause, right?
That's right.
So I think we did it. Maybe we should put this wheel away.
I don't know. Well, maybe just put it back under the table.
It's so dusty under there, though.
Well, we'll dust it next time we need it.
Okay.
It's okay.
Okay, I'll keep that in mind. dust it before we spin it.
Please.
Next time on the lab report, I call in from the beach.
Oh, come on. Another vacation?
Kids get vacation. Parents, they just supervise.
You've been listening to the Lab Report. If you like what you hear, please subscribe to our podcast, rate us, and leave us a review. To learn more about Genova Diagnostics, visit our website at gdx.net. There you'll find information on specific testing, educational resources, and how to connect with our show. Call us at 1-800-522-4762 or email us at podcast@gdx.net.
Speaking of like that Indiana corn-fed thing, do you like creamed corn?
Ugh, no.
Oh my God, I love creamed corn.
No, I don't like the combination of sugar and vegetables.
You just eat the corn right off the cob?
Yeah. Savory, butter, salt.
Okay.
Don't put sugar. Stop putting sugar in your vegetables. It's gross.
It's good though.
It's gross.